I'd like to thank everyone for coming and to welcome both the in-house audience and the online audience at UMB, Howard University, and the Department of Health. For those of you who are here, there will be pizza afterwards in the gallery, and Nicole has your tickets. So remember to get your tickets from Nicole. So it is my pleasure to introduce Dr. Anthony Leung today. He is in the Department of Biochemistry and Molecular Biology here at the School of Public Health. And his research really focuses on understanding PARP and biomarkers of PARP inhibitors and beyond. And PARP inhibitors are really becoming kind of a preferred treatment in many types of cancer. So I'm excited to hear what he has to say today. Thank you. Anthony.
Thank you very much for coming, and good afternoon. Thank you for the kind introduction. And I want to see this as an opportunity to talk to different people in the school and also outside. So I put down some of my communication method. You can come walk to my office. I'm on the 8th floor, or you can email me, social media, all are welcome. Today, I'm going to tell you about what my lab is working on. My lab is all about how genes are regulated in normal and also disease conditions. And many of you may have heard about how genes are regulated through classic nucleic acid like DNA and RNA. What I'm going to introduce to you is a non-classical class of nucleic acid called poly-ADP ribose. And some of you may have heard about poly-ADP ribose because in the context of cancer, the enzyme that makes poly-ADP ribose polymerase, short form as PARP, PARP inhibitor already FDA approved for multiple cancers, including breast, ovarian, prostate, and recently also there are results showing promising results in the pancreatic cancer as well. But although this nucleic acid is therapeutic, very important, it has been much less understudied because of the lack of tools about the molecular mechanism. And that's what my lab come into it. So what I will share with you is about some of the tools that developed in my lab and how we think that they could potentially be used for serving as biomarker. So today's talk will be very simple. We'll divide it in two parts: a basic introduction about ADP ribosylation, this is how to add the nucleic acid onto protein, and then the enzyme, the inhibitor. And then I will talk about what is known about what are the biomarker for what population, patient population, and what can be for the future. So before I start, maybe I will just kind of, because it's a school audience, and also podcast elsewhere. So let's look at this picture. Anyone know who he is? Then some clues. He's one of the founding members of the school. Any volunteer? There's a name on the book that he's reading. So he is the Elmer V. McCollum, who is the first chairman of our department of Biochemistry and Molecular Biology. So if you go up to the 8th floor, there's a McCollum reading room. That is our founding father. Actually, he is also our department is one of the first department of the school. So he's really literally one of the founder of the school. So what he's famous of is about vitamin. He was called by the mass media at that time is Dr. Vitamin. And my story is a little bit linked on this because what I'm working on, the poly-ADP ribose, is actually linked to vitamin B3, one of the vitamins that he identified. And that's another name is called nicotinamide. And poly-ADP ribose is made from compound made from nicotinamide called NAD+. And it can conjugate, like take out the nicotinamide and then conjugate it to protein. And this continue to process, the ADP ribose could continue to make like a long chain and also attach to a protein. And if you look in this chemical structure, it's very similar to other nucleic acid that you may be familiar with. Here I'm showing an RNA, which is a poly-A structure, and they share the same base. There's phosphate, there's ribose group, so that's chemically very similar. But yet it's also very distinct in the sense that it's not just a nucleic polymer unattached to other things. Here it's attached to the protein. So, and this protein modification, we call it ADP ribosylation. So what I'm going to share with you is part of my lab research is actually summarize most of my 90% of my work in one slide. So this is about tools developments for my last 8 years at Hopkins. So we developed proteomics tools to identify the modification sites. Because if we think about anything about protein modification, some you may have heard about it, it's called phosphorylation. This adding the phosphate group onto the protein. The things that you want to understand the molecular mechanisms to identify the sites. Although ADP ribosylation is identified over 56 years already, until like last few years, we don't have a method to identify where it's modified. So it's very difficult to get down how this modification works. So for the last few years, my lab developed a series of tools to identify the sites of the modification. And we'll share how we think that we can use these tools to potentially for biomarker as well. And then the informatics, we have developed a database to collect over 50,000 entries of what is known about which protein are being modified at what conditions. And then recently, we also have a first labeling tools that allows us to do a lot of study on this therapeutic important nucleic acid. So to introduce this modification, it is regulated, the synthesis is regulated by a family of 17 enzyme members in humans. They all share a domain called PARP domain, which is highlighted in green here. And although this family has known for maybe about 20 years, not much has been studied. But the one that has been studied is called PARP1, and it has been actively targeted by the pharmaceutical company. So far, it's already have 263 clinical trials, I just checked today. And the reason why it works is because that it is known that this PARP1 is important for repairing DNA. So how it works is that when there are DNA break, then PARP1 will sense where the break is and bind onto it. And then it will take it into the NAD to form a polymer onto itself, which is the major target, but also other protein nearby. Could be the DNA repair factor. And this will arrange to regulate a series of different DNA repair pathway. So if we look closer how this works is that when there is DNA damage, this PARP1 take into NAD to form this polymer of nucleic acid. And could be, you can think of like a tree suddenly form, and recruits a lot of protein to bind to fix the DNA. And as we study this more, it seems to that different protein bind in a length selective manner. So this potentially could be a timer to recruit different protein to fix the DNA at different stages. So why people really care about there are so many protein modification in cells? It's because of the two back-to-back study in 2005 that showed that this PARP inhibitor, if you can inhibit PARP, particularly for cells, cancer cells that have defects on a DNA repair gene called BRCA, then it can selectively kill the cancer cells, but not the normal cells. And as we know, BRCA has been, which stands for breast cancer susceptibility genes. This has been a major problem for a subpopulation of particular women. And if you have this modification, the sad news is that by age 80, you have 80% chance to get ovarian cancer. And one of the iconic person that you may heard about is Angelina Jolie. Her mother was died from ovarian cancer, and then and because of that, at quite a young age, and because of that, she went through genetic screening, and she also identified herself to have this type of mutation. And so the current treatment at that time was to do a preventive measure. You need to do the double mastectomy, remove both of your breasts, and then also take out the ovary so that you don't have much hormones. And then this would prevent the cancer to progress or to occur. But then now we may have a drug treatment for this subpopulation of the patient. So to understand, to explain how this PARP inhibitor work, I take advantage of the mass media. I take a video from BBC, who usually broadcasts a Christmas video, education video about science, and PARP inhibitor was featured a few years ago. But I must warn you that there is some British humor, so I'm totally not responsible about that. So the basic idea behind is that if there are cancer cells that have defects in DNA repair, so they are already very weak, and they rely all on the PARP as the remnant pathway to fix DNA. So therefore, in those cancer cells, if you treat them with PARP inhibitor, they will die. But while in the normal cells, the DNA repair pathway is still intact, if you add the PARP inhibitor, they still have other pathway to take care of it, and they will still remain intact. So the basic idea is about that. And this idea in 2005 shown in like cancer cells, animal model, that's great, 2 papers in Nature. But then it's rapidly progressed into phase 1 clinical trials, so published in 2009 in New England Journal of Medicine. And it's already showed that even in phase 1 clinical trial, which is just to check whether it's safe for patients, they already show that in some cases, the tumor even gone remission. And they have shown that this has benefits in ovarian, prostate, and breast cancer. So that's why these are the 3 cancers that people have been looked into so far. And then in 2009, that's the first phase 1 clinical trial. 2011, then it's phase 2. And here is just one of the study. Here's the ovarian cancer. You can see that this is a waterfall plot. And the bottom means that the tumor size shrink. And the top is that the tumor grow. So you can see that there's a lot of patients that's treated with this inhibitor, they have benefits about it. But what I want you to also notice is that there is a color code here. For the blue, it means that they have the BRCA mutation. And then the red one are those that does not have the BRCA mutation. So the first thing you notice is that there are more blue patients that are benefits. So those that have BRCA mutation, they have shrink the tumor. So this is what is expected. But then you also see that some have the BRCA mutation that doesn't. There's a minority of them. But even more striking, there are many patients that does not have BRCA mutation. They are also benefit. This suggesting that there is this inhibitor drug can be used beyond the BRCA mutation. And that's what is exciting about for the future, to find out who would really benefit. So this really like phase 1, phase 2 clinical trial moved very fast forward. And in 2014 already, this is the first drug that is FDA approved, the olaparib from AstraZeneca, and then rucaparib from Covis, niraparib from Tesaro, and then 2015 also there is talazoparib from Pfizer as well. And then there are totally 5 companies already competing this market. So here's the status quo. PARP inhibitor is really working for BRCA positive, mutation positive patient, but not for all of them. And even some non-BRCA patient is also benefits. This including the prostate cancer child. And then there's a lot of different FDA approval. So now the burning question is, who will benefit from PARP inhibitor? And this is an important question for 2 reasons. One is that you want to treat the patients that they will benefit. Otherwise, they would have false hope. Second is that if they are not benefit, then they will have the over-treatment, or that they will not be benefit. So you keep on treating patients that will not be benefit. So then this comes to the second part of the question that I'm interested in, is the biomarker. And the biomarker definition is something that you can measure, taking out from patients, and then to capture what's going on. Some common biomarker that you may heard of is like, for example, the lead level in the blood, to see whether the child could be exposed to some chemicals or some in the house, different surrounding, and this may cause nervous order, neuron system or cognitive disorder. The other is cholesterol level. Then if you have high cholesterol, then you may have heart disease. So can we find cancer patients that would likely responsive to PARP inhibitor? So the normal way that you can think of is using DNA sequencing, because you can use to look into the BRCA mutation and see with the patient in the cancer sample, you take a biopsy and see whether it works. And this works really well. And you already, all the approved, FDA approved indication is that most of them, they must test for BRCA mutation. But then recent studies suggesting that not only the BRCA mutation is not necessarily have benefits. I mean, you don't necessarily need the BRCA mutation to have the benefit. But also they look into the BRCA gene, because this BRCA mutation is affecting one specific DNA repair pathway called homologous recombination. So they also screen whether these cancer patients have deficiency in the homologous recombination pathway. And even they use that pathway, they also did not see a particular benefit. You can use this as a tool. So this suggesting that there is even more biomarker that can be testing what other patient could be benefits. So in 2015, there is a clinical trial, phase 3 clinical trial on metastatic prostate cancer. The major finding is that not only the BRCA gene, but also other DNA repair genes. And this what the data show here. They look into the patients, there are about 49 patients, and they find that those patients that have DNA repair defect, like ATM gene, PALB2, RED51, MRU11, this could be a range of different type of pathway. And it seems that they also benefit. And you can see that this is the survival curve. They have more than about 6 months more survival. So this is good. So then the answer could be like, well, we can just do genome sequencing on all the DNA repair genes because we know what they are. But then that's also coming to the problem. We just don't know what we don't know. Because we just have a panel to select, look into any mutation on the DNA repair genes. But what if there is some genes that we haven't studied before or characterized? Then they may not be, we may miss. On the other hand, it could be like you have the mutation, like you have the BRCA2 mutation, but you have a secondary mutation that arises that will cause resistance. And that's quite common in cancer because they keep on selecting. So these are the possible things that we need to think about when think of the biomarker. So what I have been thinking, or the field have been thinking, and I'm part of the team, is that we think that can we get closer in the gene expression cascade? Because DNA makes RNAs, makes protein. How about we look at the protein level? So if we look back into how PARP1 works, because think about how PARP inhibitor works. Again, is that when the DNA repair system is defective, then they want to rely on PARP1. And whenever PARP1 is being active, they do one thing, is to modify itself, which is the major target, but also other target as well. So could we just look into the PARP1, the ability to modify itself as a biomarker? So I go back to look into the literature. So here's what I found. So here is a Western blot to show into like what is here is probing like the beta-actin as a loading control. PARP1 look into like this, any protein level change, and also the PAR, the amount of modification. So you can see that in the BRCA2 mutation, you see that there is a PAR signal. But when you add back a wild type gene of the BRCA2, then this signal go down. So this suggesting maybe that could be a signal looking into correlation signal. And then what you can look into, another study here show that not just BRCA2. How about you look into other DNA repair genes? So here show RED54, RED52, BLM. In all these cases, compare with the control that does not lock down this gene, then you see also an increase in the correlation level. So this suggesting that this could potentially be common, because whenever you want to find a biomarker, you want to find a common one that you can potentially probe it. And this recently also shown in the ovarian cancer from Lee Cross group in 2019. This published last month. And they looked into a panel of ovarian cancer cells. All of them are derived from patients. And they probe on the ADP ribosylation level. And you can see that they just align them so that most of them, those cells that have most ADP ribosylation will be on the left and then going down on the right. And then for these cells, they also check how sensitive to the PARP inhibitor. It turns out that those that are highly sensitive, for example, these OVCAR3 cells, you can see that they are very sensitive to PARP inhibitor. It means that they will be killed. And then for the cells that have little ADP ribosylation, the HCC 5048, it doesn't change. It doesn't kill the cells. And then they also pull all the data together, and you can see there is a correlation. For cells that are highly sensitive, they have more ADP ribosylation. So my lab also looked into this, but in this case, we look into prostate cancer cells. These are all commonly available cancer cell line. They have different PARP inhibitor sensitivity, and then they have different PARP mutation. So here I just show that they have equal amounts of protein in each lane. And then here is the plot on the PARylation level. So you can see, we see it similarly, even in prostate cancer cell. But what we find is interesting is look into the particular in the DU145 cells. These are cells that is insensitive to PARP inhibitor and have a BRCA2 mutation. But when you look into the plots, they also have PARylation signal, but they are at a different position compared with the other that is sensitive. And this position likely would be the PARP1. So maybe it's about like really the automodification of PARP1 could be the signal. So this is something that my lab is really interested in. Could PARP1 could be the PARylation signal as a biomarker? So when we look into this modification, there are two things that we can think about. The first thing is the site, because this is very unique and it could be measurable. Because again, the biomarker is something that you can measure quantitatively. The other could be the length, because if you have more modification, there's two possibilities. It could be this PARP protein that have many sites that are modified. That's why you have a lot of modification. Or you have one site that is modified, but they have a huge length. So this is something that it could be looked into. But then you can ask, so do people really use like protein modification as a biomarker? And indeed, this is the case for the EGFR inhibitor. So people have used the phosphorylation because EGFR is a tyrosine kinase. And what it does is also auto-modify itself when it is activated. So we can draw the parallels that the data showing that if you have more auto-modification sites on EGFR, you will be more susceptible to EGFR inhibitor. So could you do the same for the PARP inhibitor, that you have more of the PARP modification? Then you would be good to find out the patient. And the other example that I want to point out is actually if you think about our blood type, there are three types. They are due to what kind of sugar you put on it. So this could already say that people have been doing this. But of course, there is a lot of hurdle before we get there. So one hurdle is that there, as I mentioned earlier, although this modification was identified 56 years ago, until very recently, we developed the technique to identify the sites of modification. The reason behind is quite simple if you look at this structure. This modification is very heterogeneous in length. So when you think about mass spec, they always want a unique mass signature to identify where they modify. And if you have different mass, some could be 2, some could be 3, some could be 10, 200, then you cannot find what kind of site it is. Second is that this bond here linking to the protein is very labile. Third, there's not much modification. And then fourth is that this could go to many different amino acids. So why I'm interested in the question is that I identified as a new pathway for PARP. That's how I moved into this field. But I realized no tools. Then when I started my lab here in 2011 with my brave soul first student, Casey Daniels, we developed a method that we think about this modification, and we use an enzyme called snake phenol phosphodiesterase, and then we have different iteration in the subsequent years. This phosphodiesterase has been used previously in the field just to identify what kind of structure of poly-ADP ribose is. And we think that if we chop at this between the pyrophosphate bond, the diphosphate bond, if this reaction go to the completion, then we're basically just leaving a stump on the site of the modification. And this is just a phospholipase. And it's about 212 Dalton. And it is very easily to identify on the site where it modified. And the second advantage is that there is a phosphate group. And because phosphoprotein, the way to use proteomics to identify phosphorylation sites was already very well established, we can take advantage of what is existing and then use for enriching all the phosphorylation. And then we also show that it can also enrich for phospholipase group. And then we can go for mass spec and identify the sites of modification. So this is just a very first experiment when we publish. To test, we just use an in vitro modified PARP1 and then dose into a whole cell extract so that it's a complex background, and then see whether we can fish it out. And indeed, we can fish out many different types of sites. And this is just a summary of the data, what we find. And then when we compare with data, because there are other methods developed at the very same time, so when we pull all the data together, there seems to be some consensus. For example, there are 2 sites here that could be a biomarker, because this is all common for all the activation when PARP1 is activated. Maybe we can pick this site, make some antibodies about it. After you process it, make antibodies, and then we can even have a patient, you can make a slide, detect it. That could be one possibility. Or we can use mass spec. But clinical mass spectrometry is still a way to go, but this is the future because of the sensitivity. So there's one way that we can potential. The other is look into the length. Can we use the length to measure it? And this is particularly interesting when I went to a conference and hear a talk, and they used the data from the ASCO in 2016. Here it shows a very interesting graph. So let me go through with you together. They measure 2 things when you add different concentration of PARP inhibitor. If we look into the x-axis, the lower left is the lower concentration. On the right is the high concentration. And they measure 2 parameters. One is the PAR level. So it's shown on the left. And then the other is use an assay, it's called COMET assay, to see how much DNA is being repaired or how much has not been repaired. So you would expect that when you add more PARP inhibitor, then the PAR level will go down. And then when you add the PARP inhibitor, you will have much less DNA being fixed. So there will be an increase in unrepaired DNA. So you can see that there are 2 curves here. The orange one are for one of the FDA-approved drug called olaparib, and the black one is another one from Tesaro called liraparib. So they both show very similar. Let's focus on the red one first. So you see the dotted line here going down. This is the PAR level showing. So you can see that it go down as you're adding increasing of PARP inhibitor. And at the same time, you see that another dotted line here, but then it's the circle here. It's showing that there's increasing amounts of unrepaired DNA as you add the PARP inhibitor. What interested me is the cross-section. So what is shown here at this point is that you need to go down to quite a significant level before you see a PARP, you see the repair stop to, DNA repair system stop working. So it seems that there is some transition points. So what are they? So one thing, if you look back into the mechanism on how PARP1 is involved in PARP inhibitor, is that they form different length of polymer. And this polymer length is important for recruiting different DNA repair. So maybe it's the length. If you go past certain threshold, then it becomes important. So, or in the reverse, when you treat it with PARP inhibitor, then after you past certain length, then you cannot recruit certain DNA repair. Then you start to see the DNA repair defect. So then we need to find a way to measure DNA PAR length. But then the problem is that there's no such way to do it. There's a lot of ways to doing for DNA and RNA, the nucleic acid. You can just go to the Thermo Fisher, and there's a catalog on how you can label it so that you can visualize the length. And because of this, partly of this is that this is how DNA sequencing gel look like. You can label it, and then you see where the length is, and then also the nucleotide composition is. But there's no such things for PAR. So for the last few years, we developed a new technique, which I want to introduce to you. It's called ELTA, stands for enzymatic labeling of terminal ADP ribose. This method that we developed, one of the applications that we can see the poly-ADP ribose length. But it can also do many other things, for example, to enrich the sample, like no longer we need a phosphate group to enrich ADP ribosylation site. We can use this method to enrich ADP ribose from the cells. And we picked this name, it's a joint lab effort. It's partly, this also, it means something. It means to chase in the old lost language. And we hope that now everyone can chase this therapeutic important nucleic acid. So how it works is that we can label many different types of ADP ribose, either it's a free form or it's a protein conjugated form. With ELTA, we can label it with many different type of tag, radioactive, fluorescent, biotin, clickable, many things that you can, if it is commercially available. The trick behind this is involve an enzyme called OS1, which is an antiviral enzyme present in humans. And what we did is that this enzyme has a promiscuity. That its original function is to join all the ATP together to form oligoadenylates as an antiviral signal in the cells. But then it turns out that this enzyme has also some promiscuity. It can attach ADP onto ADP ribose. So I was thinking, oh, that's a very interesting finding. But I was thinking, well, then you can label it with the commercially available ATP. But then I was thinking, well, that's not very good, right? If you can like randomly add 2 or sometimes 4 more, then you cannot really quantitate. So what I find is that you can also use deoxy ATP. That is, does not have a 2' OH group, where the ATP for the OS1 to conjugate, they require 2' OH. So in this case, then now once the DATP is conjugated to the ADP ribose, it will be a dead end. It will no longer have other DATP or ATP molecule can be extended. So we think that, oh, then because there are so many deoxy ATP available, radioactive, fluorescent, whatever, then we can just like plug and play. So the idea that we have is just mix and match. We have an ATP ribose in the middle. It could be a protein conjugated form. It could be a free form. Now we can attach multiple tags onto it. And one way that we do it is to use it is to visualize what is the length of poly-ADP ribose. To demonstrate how we do it, we can isolate the poly-ADP ribose from protein or from cells. And then in this case, we just show that we can label with P32 radioactive DATP, but we can do it with like fluorescent as well. And then we just run a sequencing gel. That's how the old days on DNA sequencing has been done. So to show that it works, we use a control. This is a bacterial PARP, HA PARP, and we know that they can make about 5 to 15 mol of PARP when people look into it, because at that time you can use radioactive NAD to visualize where it is on a sequencing gel. And then, such as a comparison, so you can see that we really get the 5 to 15 mol on this sequencing gel. And then now let's look into the cells. So we use untreated cancer cells or DNA damage cells. We know that this will induce a lot of ADP ribosylation. So you can see that in the untreated cells, we don't see any signal. And then when we DNA damage it, there's a lot of PAR being formed. There we see a ladder, so because it's a different length of ADP ribose is being added into cells. And to confirm this really is the PAR signal, so we do 2 additional experiments. One is we add the PARP inhibitor. So now, after you treated the cells with PARP inhibitor, you no longer see any of the signal. So this is really a PAR signal. And then the other thing is the PARG inhibitor. So this is the enzyme that breaks down the PAR. If you stop it from breaking down, you should have more. And that's exactly what we see here. So now back into what we see here. So can we do the same in the future, running this gel, or we can make into not just gel, we can make into a miniature device, shrink it, the whole gel apparatus into a very small device to look into whether we can do it on the bedside as well. So at this point, I just pause at this moment. Any question before I move into the next part? Happy to take a question at the intermission. Before I move on? All good? Okay. So then the next thing is that what I've been telling you is that we can use a PARP inhibitor as a single agent to treat different type of patients. Then what else, or who else can this PARP inhibitor be used? Because then you can broaden the scope of it. But at the same time, you want to find out who could be benefits. So because of a collaboration, and I'm part of a very interesting study, it turns out that PARP inhibitor can also use in combination with other therapy. Here we showcase a study with Tin Li, Wang, and also Yiming Shi in the School of Medicine. What we look into is endometrial cancer, where one of the genes, ARID1A, which is a tumor suppressor, is present in over half of the cancer. And this and we're thinking that could we use PARP inhibitor to treat them? And then what has been the general treatment is to use irradiation in the patient. And so we did a study in the preclinical state, in the tissue culture and also animal model, to look into would you benefit with PARP inhibitor? Will it be benefits with the irradiation and also combination therapy? So here's the data. It's just in a tissue culture state, just a colony assay. And the UT here stands for untreated, IR is for irradiation. And here is two different cell lines. One is ARID1A positive, which means they are wild type. The other, it's lockout. So you can quantitate how much of the cancer cells grow, or the cells that is present here grow, and after this type of treatment. What we find is that both the irradiation and the olaparib work. But what is surprising is that the two can work together in a synergistic manner. And here is some of the data that we do also perform in the mice. So here is a very good control study. There is a mouse model, xenograft. The right flank is the wild type cells, and then the left flank is you use lockout cells, and then so that they have the same exposure of everything. And what we find here is that in the mouse model, although the olaparib and the irradiation also work, the most significance that we see is when we combine the two together. And here is the data that we show that if you have the combination therapy, it can really shrink the tumor. But if you stop of the combination, then the tumor will grow back again. So one interesting aspect of this study for me is that, so can we find a biomarker as well? And when we look again on the PAR level, and you can see that the lockout cells also have an increase. So this kind of fit into our previous thoughts on like, could this be a generic one to look into not just single agent therapy, but also combinational therapy. So the use of PARP inhibitor with other agent is not new. There is a lot of concurrent project that is ongoing. I just make a summary of what is going on in the field. In addition to using irradiation, you can use genotoxic chemicals as a therapy, like the platinum drug, the targeted therapy. So like for the prostate cancer, you can target the androgen receptor. And then recently, there is also a study on immunotherapy. So this particular study published in August 2019 in JAMA Oncology is there are 2 back-to-back studies on both ovarian cancer and also breast cancer. And they find that the PD-L1 inhibitor and can also be synergistically benefit when you treat it with PARP inhibitor as well. So then that's the question. What are the biomarkers? Can you use certain things that is not just by genotype? Can it be complements to do for the future? So as a final takeaway, I hope you are convinced that PARP inhibitor is work either alone or in combination. But the question is, you need biomarker to find the right population. And then I show you different novel tools to look into the PARP. What does it work? The property of PARP, like the Leung, the site, and we have developed many other type of tools to study different properties. This could serve as a biomarker. And this not only be good for biomarker, but more I feel like is for me to understand how it works. Because if you look into the PARP inhibitor, these are those that are commercially targeted by pharmaceutical company. What I'm showing you here is a heat map on how these different inhibitors can bind to different PARP member catalytic domain. If it is red, it means that it has high affinity. If it is blue, it's low, and then the yellow is intermediate. So what we have been doing is to target the PARP1, the one responsible to DNA repair. But as you can see here that it can cross-react with many others. So that may explain what is the difference with therapeutic benefit versus the side effect, because this matters to the patient. Because, for example, the rucaparib is known to somehow cause high cholesterol. So definitely you won't want to treat with certain patient that's already at risk. So the other thing is that if we know how the PARP inhibitor work, then we also know the mechanism behind it so that we can shape it better. So if we look into the database that we created on how PARP1 work in, say here, we just look into all the sites of ADP ribosylation during DNA damage that is induced. We just use those data sets. And then do a gene ontology analysis and look what kind of pathway they affected. So we are not surprised you see the response to DNA damage similar as quite a big component, but you also see that there is many other. For example, this translation elongation is huge. So could this be also a component that we need to be careful about the RNA metabolism or how they form the complex structure? So this is something that I want to take away message for you is that PARP inhibitor not only block DNA metabolism, DNA repair, there could be more. So with this note, I want to thank my lab member who do the hard work, Casey, who developed the proteomics technique picked up by Lyle. And then for the alter technique is developed by Yoshi, Lyle, Shangyong, and also Morgan. And then our collaborator, Tini and Yiming for the cancer work. I'm excited to move to the translation work. And if there are any other people would like to do on that side, I'm more welcome to talk more. And then also Limo in Cold Spring Harbor for the alter work, and also ELAT, as well as the proteomics, our long-term collaborator, Shaoan Ang in University of Washington, as well as different funding for this particular project. And I'm here, stay here to take any question. Thank you very much for your time. Yeah, Ben.
Very nice.
Thanks.
I was curious about that assay, if you tested it at all on formalin-fixed clinical specimens, and if not, I mean, just theoretically, should that work, or are you going to have a huge amount of interference from mRNA, or what are the caveats?
So I think if we can extract from the sample, then it shouldn't be a problem because we have a way to remove DNA or RNA. So the OS one, what is known is that it could potentially add on to single-stranded RNA. So that could be one of the kind of the interference. But yeah, then that should be able to work because one of the how OS1 work is that it need a double-stranded RNA to activate it. So definitely, even if we put a lot, it will not affect if it's double-stranded form. But if it's single-stranded form, if you have a 2' OH, which is in the ribose, same in the ATP, the ribose, that might be a problem. Yeah. Yeah, sure.
I had one other question.
Yeah.
Has anyone developed a gene expression-based biomarker, like a gene expression signature-based biomarker for response to PARP?
For PARP inhibitor? Yeah, there is a study just published a week ago, and Genome Biology, they look into it, but it's not commercialized. So that is a potential signature, but that need to be in. Really go into the real test, you know. As you know, there are many kind of signature. But, yes. But then the BRCA assay is well commercialized. Yeah. Okay. Yes, Tao?
I have simple question, but also related method, the biomarker. How do you test to make sure not like false positive results? I mean, the mismatch part, you know, like if you run it down, see signals, like say, oh, at one patient got some.
Yeah.
Something wrong.
So on the gel, we may be able to tell like which protein is being modified based on the molecular weight. So that's very rough. But then in the future, if we do same size and length, then we can identify which protein is really being modified. So this gives us the specificity on what it is. What our current thought is that because PARP1, during the DNA damage, PARP1 itself is the major target. So likely, PARP1 would be the target one. We may develop further assay to enrich them, etc. Yeah. Okay. Thank you very much.